Good morning, sir.
Good morning.
May I start now?
Yeah.
Okay.
Am I audible?
Yes, sir, you are audible.
Good. Can you share the slide?
A very good morning to all of you. I once again welcome you all on day 2 of AICTE sponsored STTP on future trends in research methodology for drug discovery and development. We have with us Dr. Shekhar Verma. Before actually starting his lecture, I would like to take this opportunity to introduce the speaker to our participants. Presently, Dr. Shekhar Verma sir is working as professor and principal at Pandit Deen Dayal Upadhyay Memorial Health Sciences and AYUSH University of Chhattisgarh, Raipur, India. He has done his graduation from University Teaching Department, Guru Bhartas Vishwavidyalaya Bilaspur, and done his post-graduation from B.R. Nahata College of Pharmacy, Mandsaur, Madhya Pradesh. He has completed his PhD from Guru Bhartas Vishwavidyalaya Bilaspur, and sir has vast experience of academic, administrative, and research work. Dr. Verma is actively involved in research in field of and control of drug delivery systems, non-aqueous dispersion of poorly aqueous soluble drugs, herbal nutraceuticals, etc. Presently, he is working on new funded research project, and Dr. Shekhar Verma is nominee of CPC SEA, New Delhi. He has awarded Best Young Teacher Award by Premier Pharmacy Publication, Nirali Publication, New Delhi. Sir is life member
Gunjan sir, we can start. Gunjan sir, we can start presentation.
Okay, sure.
Are you sharing my slide?
Yes, sir.
Okay, please.
The slides are visible to you?
Yeah, it's visible. Just maximize it.
So now it is visible?
Yeah, it is visible.
Yes, sir. You can start now.
Thank you very much, Gunjan sir, for my brief introduction. And thank you very much, Columbia Institute of Pharmacy, Raipur, AICT New Delhi. You provided me opportunity to share my scientific view about the novel nano carrier system. And it's a very wonderful to see that during this pandemic, you are doing a very commendable job. You are creating such a positive impact, not only for the students, but also among the faculty members. In the first phase, I was witness of some sessions. Two sessions were very fruitful. And before I start my today's presentation, just I would like to brief about my title, the versatile novel nano carrier system for various water unstable drugs. Actually, in controlled drug delivery system, in normal drug delivery system, we have large number of carrier system that carries the various kind of drug, including water soluble, water insoluble, and other variety of drugs for the targeting the infected organ or in many areas. But what we have seen since the last the 2 decades, most of the new chemical entities which are coming into the market, around 60 to 65 chemical new entities are water insoluble, or we can say it is hydrophobic, and is not hydrophilic. So for that reason, it is very difficult to select a suitable carrier system for those kind of the drug, for the better bioavailability and for the better pharmaceutical efficacy. So based on this theory and phenomena, I will try to explore some novel carrier, especially for the water unstable drug. It means whether drug, maybe the poor aqueous soluble drug, or very drugs maybe very sensitive towards the water. Please next. Now, what's new on these presentation? What's new? Actually, before understand the theory and phenomena of novel nano carrier system, we have to know about the rationale of nano carrier system, the selection of carrier system based on drug property, advantage of novel nano carrier system for water unstable drug. Actually, I will try to explore, and I also done in my laboratory. I try to develop a dispersion system without water phase. We can say nano emulsion or traditional emulsion. I try to avoid water phase. Why I try to avoid water phase? Because, as I said, most of the new chemical entities, around 60 to 65%, which are coming into the market, are pure aqueous soluble. For those kind of drugs, we have to develop such a system which are very compatible or viable for the drug. That's why I try to replace water with the various kind of the organic solvent and polyols. Next, please. Sir, am I properly audible?
Yes, sir. थोड़ा मतलब network disturbance आ रहा है, but you are audible.
Okay. हाँ. Actually, I am in home. I am using the mobile network. Yes, sir.
वॉइस थोड़ा सा अनक्लियर आ रही कहीं कहीं नेटवर्क के कारण.
हाँ हाँ हाँ.
ठीक है ना कैरी ऑन सर.
Okay. Now, please try to understand the difference between the traditional emulsion and the anhydrous nano emulsion system. We have studied in our initial level of the graduation during the first semester of B Pharm and second semester of B Pharm that emulsion is a biphasic liquid dosage form, and it has two types: water in oil and oil in water. Okay, that definition said both two phases should be immiscible. That definition not state water is mandatory for the emulsion. They described previous author described the emulsions are the two types: water in oil and oil in water. But water is not a mandatory for emulsion. We can make a emulsion without water phase also. So in this novel nano carrier system, I will try to explore, I will try to describe about how we can make an anhydrous nano emulsion system without water phase. And these are best suitable for the water unstable drugs compound. Okay, now the anhydrous, we can use anhydrous, that is obviously the absence of the water in the one nano emulsion system. And the nano emulsion, the size range should be around 10 to the power -9. As we know, the emulsion is a biphasic liquid system in the type of solid form by combined two liquid that normally don't mix. The process of mixing liquid to form an emulsion is called emulsification. As I said earlier, actually it is a basic definition which we have studied during our graduation, and we taught many students in all semesters. The emulsion can be formulated. See, and try to understand the emulsion can be formulated without an aqueous phase to produce anhydrous or non-aqueous or oil in oil system. Such system which can replace conventional emulsion where the presence of water to be avoided. Why I used novel nano carrier system? Because in this dispersion system, nano emulsion system, I replace the water with the another kind of the solvent or any polyols. Polyols, compound which having more than 2 OH group like glycerin, glycerol, and there are a larger number of compound of this category. Please next. Now the requirement for the anhydrous nano emulsion. Actually, this anhydrous nano emulsion is a novel term. Students, researchers, and the scientists, the scholars are studying and doing a deep study, deep experiment inside the laboratory about the nano emulsion, about the micro emulsion. in all that cases, the water they are adding water. But here the requirement for anhydrous nano emulsion is we will avoid to use the water, because in the conventional definition of emulsion is not saying the water is a mandatory for the emulsion system. So you can see a slide. The requirement for the anhydrous nano emulsions are the surfactant in the combination of surfactant, non-aqueous polar solvents, or polyols, oil phase. See, there is no water phase. It is a novel. It is a novelty of this system, avoiding the water. And the surfactant in the combination of surfactant. We have various types of the surfactant: anionic, cationic, non-ionic, amphoteric, and various kind of the polar solvent that is immiscible with the oil, and the various types of the oil phase. These are very basic requirement to prepare the anhydrous nano emulsion system for the entrapment of water unstable drug or water sensitive drugs. Please. Why need to develop anhydrous nano emulsion? The first question it is in mind when students and researchers or faculty members think about, we have already familiar with the different type of stable and the popular dosage form. Why need to develop the anhydrous nano emulsion system? Because as I said in my earlier slides, that approximately 65% new chemical entities are hydrophobic in nature. It means water poor aqueous soluble, and around 65% drugs. To protect water unstable and water sensitive drugs, to make water free dispersion system for various types of water insoluble drugs, avoid phase separation associated with traditional emulsion, to get a stable system for extended period of time, to maintain globule vesicle droplet size. So these are the few reasons where we can see why we have to develop these kind of the anhydrous nano emulsion for the better bioavailability for the large amount of the water insoluble drug or water poorly water soluble drugs. And obviously when we entrap anything into the vesicles or globule, definitely that the outer layer or spherical shape or globular shape will be definitely protect the drug. These are the few advantages. That's why we have the large number of advantages part of this system, and definitely it will be a miracle in the science of the control regulatory system. Next, sir, please. Now the advantage of the anhydrous nano emulsions. See, I have already discussed the few advantages during the introductory slide. The anhydrous emulsions are suitable for poorly soluble drugs and thermodynamically stable multi-component fluids. The avoidance of phase separation. These are phase separation is very common in all types of the dispersion system. Traditionally, we added emulsifying agent, but it is a very short-term and not a permanent solution to make a stable formulation with the help of the emulsifying agent. And to avoid these phase separation, to get the proper stable dosage form, we have to select the specific suitable surfactant or combination of surfactant to get the stable formulation. Increase the drug loading capacity, carrying capacity of both lipophilic and hydrophilic. These 4th point is the most important point, because these type of the anhydrous nano emulsions have capacity to carry both the lipophilic and the hydrophilic drug. Even the plant extracts can also be easily deliver globules or vesicles prepared by this emulsion system. And the protection against intestinal protease is a very common advantages of this kind of system. Increase the bioavailability of the drugs, easy to formulate and prepare. Now the rational, or we can say the basic strategy in based on the principle, we can make the anhydrous emulsion. There are two basic strategies. Two basic strategies could be considered when searching for stable anhydrous emulsion system. Number one, to design surfactants having two incompatible blocks, each of which is actively soluble in either of the immiscible liquid. It means we have to design, we have to fabricate a novel type combination of the surfactant that should be compatible blocks, and which is selective soluble in either of the immiscible liquid, either oil or polyols or organic solvent. See, here we are not using water. We are replacing water with the polyols or organic solvent. And the second strategy is, the other approach is to search for a suitable oil immiscible polar liquid that can substantially replace water using emulsifier. This technology is based on the polyol in oil emulsion that is stabilized by the different type of surfactant, combination of surfactant and surfactant precursor. That's why we call it surfactant based nano emulsion or nano carrier system, because when we design or when we fabricate various mixtures of the surfactant, then we can definitely get the proper emulsification. And how we can make a proper emulsification? See in the present slide, the emulsifying effect of various surfactant on the stability of glycerin, olive oil, and hydrous system. See, just I am showing the one example. I took one agent that is polysorbate 20, that is basically twin 20, and I used different concentration of this 1.5, 3%, and 5%. Method is emulsification. And which type of emulsion appeared? Here, separated, here, separate, here. When we use two immiscible phases and various types of the mixture of surfactant, there will be the emulsification method or separation method. If emulsification done, it means both phases are easily mixing together with the presence of the surfactant. And appearance, definitely appearance should be clear. It should be not separated. If it is clear, that means we can go for the further study of that kind of the sample. In the second example, you can see that it's 60 is a polysorbate 60. Again, just I'm showing only for the example of these two types of the agent, that is a surfactant twin 60 and twin 20. And the when method apply for the mixing, if a separation is done or separation completed, it means our formulation has been failed. If emulsification is formulation is successful, but appearance should be also clear. So based on the emulsifying effect of various surfactant on the stability of glycerin, olive oil, and hydrous system, it is a one type of system. Glycerin is a basically polyol, so we can use glycerol also in the place of the glycerin, and the olive oil we can use a castor oil, eucalyptus, any kind of the oil. There are we can make various types of the combination of the different surfactant, and we can observe the emulsification method and found which formulation is suitable. It's suitable means which is stable for the extended period of time. Please. Okay, just I'm trying to showing some how we can prepare this nano anhydrous emulsion system or anhydrous nano emulsion system. Graphically, you can see that the very simple method. With the help of the sonicator, we can make this kind of the system. With the help of the magnetic stirrer, we can make this kind of system. Homogenizer is also one promising approach to prepare anhydrous nano emulsion system. We'll go for preparation into the laboratory level. Definitely, we have to select two suitable phases, that is the polyols or organic solvent, and the second phase is definitely oil. And the third, the most important part is we have to design the surfactant or various types of the mixture of the surfactant that will be efficiently stabilize the system and shows the proper emulsification and proper stability without the separation of phase separation for the extended or maximum time period or the extended period. Okay, so here I'm just showing the magnetic stimulus, but the sonicator, both the sonicator, probe sonicator and the mass sonicator are very much suitable to make a smallest size of the system. And ozone is also the most promising approach. Sir, next, please. Sir. Okay, now the most important part of any kind of the presentation or any kind of the exploring the new theme, that is the application of versatile vehicles. Application is the most important one, because the everything if our manufacturing process or our or the evolution parameters, okay, then we have to find out applications of the system. Applications mean always it would be suitable, it should be economic, it should be compatible, it should be biocompatible, it should be compatible for the drug also. To see the water-free liquid preparation and number of drugs. Most important, water-free liquid preparation and number of drugs. Versatile vehicle for poorly aquasoluble drug. It has